Today we're going to review most of the things that we've talked about so far in this class. You've probably noticed quite a bit of overlap between how cells die in different diseases, because there's really only so many ways that you can kill a cell. Now it might seem like there's a lot, and I get that the illustration shown over here looks a bit crazy, but when you get down to it, there's only about 2 ways to kill a cell. Really there's 3, but we'll say there's 2. Apoptosis or necrosis. Now how that comes about, well, that depends on the condition. So we're going to go through a few different ways that cells die that we see commonly in different neurodegenerative diseases. We'll start off with glutamate toxicity. With glutamate toxicity, this is very much related to excitotoxicity. Neurons get hyperactive and that leads to their death. As far as glutamate receptors go, there's really only 2 that we need to talk about: Ampa and Nmda. A great way of distinguishing these is to think about their IV relationship, or the amount of current, that's the I, that they generate based on V, the membrane potential. Now for Ampa receptors, it's a nice straight line. They're going to generate an electrical potential based on the cell's charge, and that's about the only thing that matters. If we have a negative membrane potential, let's say we're at rest, somewhere about -70 or so, we're going to have an inward current. We're going to let positive charge flow in. That should make sense. If we have our negatively charged cell here, it's negative on the inside, that's going to attract positive charge inward. At positive potentials, we'll get an outward current. So if we hold the cell at, let's say, positive 50 millivolts, well, that's going to be repulsive to positive charge, and we'll see it flow out. So Ampa receptors are going to have this nice straight IV curve. Nmda receptors, on the other hand, have a typical J-shaped curve, where near resting potentials you don't see much of a current. And we'll talk about why that's the case next semester. But for now, what we need to realize is that when neurons are communicating under normal basal conditions, let's say here's a nice dendrite, and here's the axon. When we release glutamate, this is primarily going to stimulate Ampa receptors if the neuron is resting. And only if the neuron is already depolarized are we going to stimulate Nmda receptors. Now, the difference between Ampa and Nmda receptors lies not only with the shape of their IV curve, but also how many ions are they going to let in. So, across time, here's what our currents are going to look like. Let's say here's no current. Ampa receptors are going to have a very fast but rapidly desensitizing current. The Nmda receptors open a little slower, but they stay open for much longer. So when you think about the area under the curve, that's related to how many ions they're letting in. And Nmda receptors let in lots of ions. The ion that we're thinking about here, calcium. Both receptors can let in calcium. There are some Ampa receptors that are calcium impermeable, but there are no Nmda receptors that are calcium impermeable. We're going to let in a whole lot of calcium. That'll come up again later. Now, when everything's working just fine, glutamate release is not a problem. But when neurons are hyperactive, then we have an issue here. Now, what's going to cause neurons to be hyperactive is variable. It could be from ischemia. So if we have a decrease in blood flow and we don't produce enough ATP to fuel our sodium-potassium pumps, that's going to cause depolarization. So if you look at the figure on top there, that's showing you depolarization of a neuron after the addition of a sodium-potassium pump inhibitor. So when they add 1 micromolar wabane, that's the inhibitor there, you'll notice that the frequency of action potentials increases and the membrane potential rises up. It depolarizes there. Eventually, we reach what we call a depolarization block. The cell doesn't fire any more action potentials. And if this isn't corrected, the cell will die. So it could be from a lack of ATP. It could also be from reactive gliosis. So if we have something damaging the nervous system, that can cause reactive gliosis, where astrocytes are going to modify their function. One of the things that astrocytes are going to do is pick up potassium so it doesn't build up. Because if potassium builds up outside the cell depolarizes. And that's what the plot on the bottom is showing you. So as you increase extracellular potassium levels, the membrane potential is going to depolarize. That's going to make it easier for neurons to fire action potentials, and every time a neuron fires an action potential, it spits out glutamate. When it fires a bunch of action potentials, it spits out a bunch of glutamate. Now, of course, this gets picked up by astrocytes unless they're undergoing reactive gliosis. There's some evidence that there's an increase in glutamate transporter expression. There's evidence that there's a decrease. So the reactive gliosis could impair not only potassium reuptake but also glutamate uptake. Both of those are going to lead to depolarized neurons, and depolarized neurons are going to spit out more glutamate. Glutamate's also going to come from the reactive microglia. Any white blood cells that have infiltrated the central nervous system, for example during infection, and tumor cells will also release glutamate as well.
Sorry, I was being angry.
That's just showing us the lack of potassium or glutamate reuptake. So, what happens as a consequence? Well, when neurons are hyperactivated, when we spit out a bunch of glutamate, remember the whole purpose of releasing glutamate is to create an electrical potential in the postsynaptic neuron, and that electrical potential is driven by the movement of ions. So as we release this glutamate here, positively charged ions are going to flow into the cell and cause it to depolarize. Now as we have an increase in ion concentrations, osmosis is going to occur. So water is going to follow the movement of ions. Of course, this will go in through aquaporins. Those are just water holes, that's all that means. And as the cell fills up with water, what this is going to cause is a little bit of swelling. And if we continue to excite the cell, bring in ions, bring in water, it's going to eventually pop. So it'll balloon up too big, pop, and there we have necrosis. So that's one of the ways that we can kill cells in an uncontrolled manner. That necrosis is going to release a whole bunch of PAMPs, a whole bunch of DAMPs, damage associated molecular patterns, and that damage there is going to then stimulate inflammation, further modifying astrocyte function, recruiting white blood cells, causing an excess of glutamate release from them. And that's the reason at the beginning of this talk I had all those arrows pointing to everything, because it's all connected. Now, assuming that we don't experience necrosis, we survive that, and most of the time we will, we run the risk of apoptosis. Because as we're moving ions in, we got to pump them out. That, of course, isn't free. We're going to be spending ATP. And anytime we burn up our ATP, that means we have to synthesize more. ATP synthesis always runs the risk. There's about a 1 to 2% chance of creating free radicals, or reactive oxygen species. Those free radicals will then damage the cell in some non-specific way. Now, if they damage the mitochondria and cause it to permeabilize, or if we have an excess of calcium floating around, that calcium is going to create what we call a mitochondrial transition pore. So the mitochondria are going to try to suck up that calcium. When we permeabilize the mitochondria, stuff leaks out. The stuff that I'm talking about here would be components of the electron transport chain like cytochrome C. And cytochrome C is going to stimulate caspases. And those caspases are what carry out apoptosis. So it's going to be controlled cell death. But either way, the neuron's gone. Now this can also be triggered by the leakage of glutamate outside of the synapse. If we have an excess of glutamate release and it floats away, it can stimulate extrasynaptic Nmda receptors. So let's have a look at those. Now normally we're going to buffer the glutamate after it's released. So we should have astrocytes surrounding the synapse, helping to pick up that glutamate. So here's the astrocyte, here's our presynaptic site, here's the postsynaptic site. Now so of course we have our Ampa, we have Nmda receptors as well. We also have extrasynaptic Nmda receptors. And these are a little different than those you find at the synapse. If we have the leakage of glutamate away from the synapse, which we will if we have a burst of activity, so if this neuron is hyperexcited and releasing an excess of glutamate, or if we have an impairment in our ability to pick up glutamate with our excitatory amino acid transporters, so that reactive gliosis that impairs glutamate uptake. One or both of these is going to allow glutamate to stimulate extrasynaptic Nmda receptors. These are generally cytotoxic. What they're going to do is stimulate the expression of pro-apoptotic proteins, such as BH3 only proteins, and what these do is facilitate permeabilization of the mitochondrial membrane. So they're going to create little holes in the membrane there that are going to allow the leakage of those other pro-apoptotic factors, then triggering cell death. So there are many ways that glutamate can kill the cell. The next topic is protein aggregation. So we see sticky proteins in a variety of disorders. We see those amyloid plaques, we see neurofibrillary tangles in Alzheimer's disease, we see Lewy bodies in polyglutamine disorders, we have intracellular aggregates. They're all over the place. And they're all doing basically the same thing. They are impairing the normal function of the protein because it's now pulled out of solution, and they're creating a big ball of crap that the cell has to deal with. So in the central dogma we go DNA, RNA, protein. And it sounds like it's just a nice two-step process. Turn DNA into RNA, turn RNA into protein. But of course, once we make RNA, we have to edit it, we have to process it. Once we make a string of amino acids, well, we got to make sure it folds up properly, because there's many ways that that little bead of amino acids could be folded up, and structures function. So it's really important that that bead of proteins gets folded up properly. So we have a large host of proteins that are going to ensure proper protein folding, and this would be our proteostasis network. Think of it as a two-part system. One part would be the chaperones. So chaperone proteins are going to assist with protein folding. they're going to bind to certain parts and make sure that it folds up properly so that we're left with a nice, happy, properly folded protein. Chaperones are going to carry that out. That's when things go well. When things don't go well, then we have the proteasome. What this is going to do is destroy proteins that won't fold up properly, or damage proteins that have been tagged for destruction. So ideally what we do is fold up our proteins properly and they go do their job. When proteins don't fold properly, we get rid of them. The problem with aggregation-prone proteins is that they form these misfolded balls and they're very difficult to break down. So what we create is a big pile of junk. Usually these are going to have a bunch of beta sheets in them. So big flat structures that allow proteins to just stack on top of one another. So this is going to be our aggregate. Can't fold that up. What we're going to try to do is break it down. And so components of the proteasome are going to bind and try to destroy it. But they won't be able to. So then the other proteins, as they wear out or as they misfold, they don't get broken down. So what we start to do is decrease the proteasome function of the cell. So that all the rest of the proteins in the cell aren't being properly destroyed in the right time. And we can see the buildup of proteasome here. So the green signal is showing us polyglutamine, so big balls of junk. And what the red is showing us would be components of the proteasome. And you'll notice that the red and the green have a very similar pattern. So part D and E are the same image. So just imagine them laid on top of each other. G and H, same image. Just imagine them on top of each other. So where we have big balls of green, or polyQ, we also are sequestering our proteasome. That leaves all the other proteins free to misfold and create problems for the cell. Now, the proteasome is going to also be involved with destroying the mitochondria. So it's going to help tag it for destruction so that it gets engulfed by lysosomes. If we're tying up our proteasome by trying to destroy this ball of junk, what that allows is the accumulation of damaged mitochondria. Now, as we just discussed, if mitochondria are damaged, stuff's going to leak out. And the stuff that we care about here would be pro-apoptotic factors like cytochrome C. And we're back to where we started. That's going to trigger apoptosis, and the cell is no longer with us. Whenever we impair proteasome function, we undoubtedly see signs of mitochondrial dysfunction. Because mitochondria, just like everything else, wear out. Part of normal cell function is detecting the organelles that have been worn out, tagging them for destruction, and then making sure that they get engulfed and destroyed, in this case by the lysosome. If we can't do this part right here, if our proteasome can't tag the mitochondria as being damaged, the cell doesn't destroy them. They don't get engulfed by lysosomes, and they're free to leak out pro-apoptotic factors or to allow for the production of free radicals, because parts of the electron transport chain are gone. So those electrons moving through the mitochondria have nowhere to go. They don't have a proper acceptor, so what they do is leave and create free radicals as opposed to free water. That leads us to mitochondrial dysfunction. You probably think of the mitochondria as the powerhouse of the cell, because that's probably what you heard in your high school biology class, and that's totally true. They are also calcium sponges, and they're also regulators of cell death. So they're also the self-destruct button for the cell. But this is primarily what you've learned right here, powerhouse of the cell. The way that they're powering the cell is with the electron transport chain. So we'll break down usually carbohydrates, and since we're talking about neurons, almost exclusively carbohydrates, in order to create high energy molecules that will then donate electrons to the electron transport chain. This is showing us the electron transport chain there. You'll notice cytochrome C is labeled right there in the intermembrane space. And so that's one that's free to float out of the cell when the outer membrane is permeabilized. Now, the electron transport chain is a wonderful system. It's going to, if I can simplify it for you here, it's going to move electrons through. Those electrons are going to power proton pumps. So we create a proton gradient, and that proton gradient is going to flow out through an enzyme called ATP synthase, and that's going to trigger the production of ATP. And this is how we power the cell. In order for this whole system to function, electrons have to keep moving through the electron transport chain, eventually bumping into a couple of protons and a half an oxygen molecule. Just to keep the formula simple, we'll throw 2 electrons through there and we end up with 1 water. If electrons can't move through the electron transport chain, either because we don't have an acceptor or because we've had mitochondrial damage, then the electrons flow out of the electron transport chain and make free radicals. We might run out of oxygen if we experience ischemia, for example. So in the case of stroke, when we have a blockage of blood flow to regions of the brain, that blood doesn't enter, doesn't bring the oxygen, and the oxygen that's there is quickly used up. And without oxygen there to accept electrons, they're going to go to something else, and that something else is going to be a non-specific component of the cell. Of course, we've already gotten the connection between protein aggregation and mitochondrial dysfunction. There we're failing to tag damaged mitochondria. We could also have specific mutations that disallow the tagging of damaged mitochondria. So there's 2 proteins that we got to talk about here, and these are commonly mutated in familial cases of Parkinson's disease. And that would be PINK1 and Parkin. PINK1 is going to be the protein that tags mitochondria as being damaged. Now in normal, healthy mitochondria, PINK1 is going to bind to the outer membrane there, get taken in, and degraded, pretty much. That's when it's healthy. Now in the case of damaged mitochondria, we can't take in PINK1, so PINK1 builds up on the outside of the mitochondria. That's going to tag it as being damaged. PINK1 has a binding partner named Parkin. Now Parkin isn't going to bind to healthy mitochondria because they continually bring in and get rid of PINK1. But when mitochondria aren't working properly and PINK1 has built up, then Parkin binds, and what Parkin does is tag the mitochondria for destruction. What it does is ubiquitinate it. It adds a bunch of ubiquitin molecules on. And that's taken as a sign that this is damaged goods. So when we ubiquitinate proteins or organelles, that is going to signal to the lysosome that this is damaged and needs to be engulfed and destroyed. This way we don't allow damaged goods like the mitochondria to accumulate in the cell. Because if damaged mitochondria accumulate in the cell, they're going to stimulate apoptosis. We don't want that. Whenever we have mutations in PINK1 or Parkin, we don't properly ubiquitinate damaged mitochondria. They build up, and cells like dopamine neurons that are susceptible to oxidative stress and mitochondrial damage are going to die. So that's the connection between PINK1 and Parkin. It all comes down to mitochondrial dysfunction. Whenever we have mitochondrial dysfunction, same thing happens as we've already talked about. Oxidative damage to the cell through the release of free radicals, triggering of apoptosis through the leakage of cytochrome C, and we're also not producing as much ATP. If we don't have enough ATP to fuel our pumps, we depolarize. And if we depolarize, we might experience excitotoxicity, either through necrosis or through further causing metabolic stress and leading to apoptosis. Inflammation is going to occur anytime we have damage or infection. So those DAMPs, those damage-associated molecular patterns, or the pathogen-associated molecular patterns, those are going to get picked up, and those are going to get picked up by microglia, and that's going to trigger an immune response. So the microglia are going to be kind of the first line of defense in the central nervous system. Inflammation can also occur without overt damage in the case of brain tumors. So tumor cells can release pro-inflammatory factors like tumor necrosis factor. And different tumor lines are going to release different amounts of tumor necrosis factor. In some cases, TNF seems toxic to tumor cells. In other cases, the tumor cells are releasing it themselves and it doesn't seem to bother them. It's a fairly complex story there. But tumors can indeed release TNF, and if they do that, that's going to trigger inflammation, and that's what these data are showing us here. So we're just looking at the essentially the amount of TNF alpha mRNA. So what they did was isolate mRNA. They did reverse transcriptase PCR to create cDNA because it's more stable, and that just allows us to see what genes are being expressed. Because every cell has TNF alpha in the DNA, but only cells that are creating TNF alpha will make TNF alpha RNA. So that's why they're looking at that instead of DNA. Anyway, if you just look at the bar graph on the bottom and not worry about all that stuff, you can see that in ovarian tumor cells, compared to normal ovarian cells, we have an increase in the amount of TNF alpha RNA. So tumors are creating and releasing TNF alpha, and when they do that, they're going to trigger an inflammatory response. So it could be from damage, it could be from infection. That infection could be some pathogen, or it could be an invading tumor cell. And that tumor cell could also be a primary tumor cell that started there in the brain and has just mutated over time. What inflammation is going to trigger is reactive gliosis. This will do all the same stuff that it did in the first part of the talk. So we're still going to mess with potassium reuptake and glutamate buffering. What we're going to see here is this kind of positive feedback loop between microglia and astrocytes. So once we trigger inflammation, so when the microglia here and the astrocyte sense damage, microglia are going to pick this up first. That's going to trigger the release of TNF alpha. That will stimulate reactive astrocytosis, and reactive astrocytes are going to release TNF-alpha, as well as other pro-inflammatory cytokines. So there's more to it than just this, but just to simplify the story a bit, this positive feedback loop here is going to maintain reactive astrocytosis and keep our microglia reactive. So until we deal with whatever problem is causing the inflammation, this positive feedback loop keeps our astrocytes and microglia in a reactive state, so that they can then seek out and either engulf and destroy the pathogen, or remove the little bits of damaged cell, or trigger an inflammatory response so that we can increase the diameter of our blood vessels, increase the permeability of the blood vessels, and allow the entry of white blood cells. And these can help deal with the infection and damage. Of course, they'll also release glutamate. That's not a good thing. And the fluid that comes out with them can lead to swelling. That swelling can then affect local blood flow, leading to ischemia, which leads to depolarization, mitochondrial dysfunction, etc. So it's a tangled mess, in other words. Now, all that stuff that we've mentioned up until now has just been repetition. We've heard all that before. I want to introduce something a little new here, and that would be alterations in neurotrophic support. When we're talking about neurotrophic factors, that's just a way of saying things that nourish neurons. So neuro would be neuron, trophic meaning that it feeds it or promotes the health of it, and factor is just how we say thing when we don't want to say thing. You've heard use it or lose it before. That's still true. So when we use it, or when neurons fire an action potential, this is of course going to propagate down the axon, but it'll also move into the dendrites and cause the release of neurotrophic factors that's going to help keep the presynaptic neuron nourished. So when this neuron fired some action potentials and stimulated the postsynaptic neuron, if it was successful, and this neuron fired an action potential, it gets a little treat. It gets a little retrograde message to help keep it alive, and that would be a neurotrophic factor. Primarily, neurotrophic factors are going to be acting in the axon. What they do is bind to neurotrophin receptors, and these come in a few different varieties. They're all going to be dimeric receptors, so there's two subunits there. And we have two general classes of these. We have our TRAC receptors. These are good. And then we have our P75 neurotrophin receptor, and we generally think of this as bad. This is an okay oversimplification. Of course, it's not perfect. But neurotrophins can bind to both. It really just depends on the concentration, or whether they've been processed properly. So we always create our neurotrophic factors as a pro-neurotrophin, and then that gets cut into a smaller active neurotrophic factor. The unprocessed ones bind much better to the toxic receptor. The fully processed ones bind much better to the trophic receptor. At high enough concentrations, though, the processed neurotrophic factor can still stimulate the toxic receptor. So if this cell is going to be nourished, it has to get the receptor into the axon allow it to be stimulated, and then transport it back. So the first thing we have to do is make the receptor. Now, of course, that involves creating RNA, moving that RNA out of the nucleus, and creating a protein. This process seems to be impaired in cases of Huntington's and ALS, at least in some cases of ALS, because of mutations in proteins that handle RNA. So if we don't properly shuttle RNA out of the nucleus, we're not going to turn that into protein. If we don't make high enough levels of our neurotrophin receptors, neurons won't be nourished, and it doesn't matter if you use it, you're still going to lose it. So this illustration is just showing us two different receptors. You got your blue track B receptor, that's going to bind BDNF. This is one of those neurotrophic factors. That just stands for brain-derived neurotrophic factor because we discovered it in the brain. On the right, there's the red P75, or the toxic receptor there. You'll notice right under it, it says apoptosis. And what the track receptors are going to do is stimulate survival by inducing anti-apoptotic gene expression. So we got to get these things all the way back to the nucleus because we put them out there in the axon. That's what this illustration is showing us here. So we got our cell body that has the nucleus. We're showing us that in A. Panel B there is showing you that we're having that retrograde release from the dendrites to the axon for our neurotrophic factors. And then that third panel is showing you that down there in the axon, what we're going to have to do is internalize the receptor and transport it. So we're going to have to pull it inside a vesicle with its neurotrophic factor. And then we got to run back to the cell body because this is out in the axon. So we're going to need some retrograde transport. That means anything that impairs axonal transport can impair this process too. So things like tau accumulation, or in these data, alpha-synuclein, can lead to impaired neurotrophic support. So even if we're making our neurotrophin receptors and they're getting stimulated, if we can't bring them back to the cell body, it doesn't matter. So in these data, we're seeing impaired retrograde transport with alpha-synuclein. So we got our chymographs, just like we've seen before. So as you go from top to bottom, anything that stays as a straight line means it didn't move. And anything that's shifting over to the right means it moved. So here's the beginning, here's the end, and they're looking over 120 seconds here. This thing didn't move. This thing moved. This thing didn't move. This moved a little bit and stopped. So as we're shifting to the right, that's showing us what we want to see. That is our retrograde transport, which is what has to happen if neurotrophin receptors are going to prevent apoptosis. You'll notice the difference between the top and the bottom there. The top is just normal, old, and non-transgenic neurons. Nothing special about them other than they've had their picture taken. In part B there, they're showing you the transgenic neurons that express alpha-synuclein. By expressing higher levels of alpha-synuclein, they have impaired axonal transport. So those Lewy bodies and the alpha-synuclein that makes them up, what those are going to do is, in this case, impair axonal transport. And that interferes with neurotrophic signaling. So now you can use it, but you'll still lose it because we're not stimulating the anti-apoptotic gene expression in the nucleus. Because all that stuff happens in the cell body. Now, I hope you remember the concentration difference between activating TRAC or P75. Let's see. So the TRAC receptors, these are stimulated by low concentrations of our neurotrophic factor. And that is going to be nurturing to the cell. At high concentrations of our neurotrophic factor, then we start to stimulate P75, and that is going to trigger apoptosis. The release of neurotrophic factors depends on the level of activity of the cell. So this takes us all the way back to the beginning when we talked about glutamate toxicity or excitotoxicity. So when neurons fire a bunch of action potentials, that hyperactivity, yeah, it's going to spit out a lot of glutamate. That can be toxic. It can be toxic to this cell too with all that ion movement that's going on. But it can also be toxic retrogradely by spitting out too much neurotrophic factor. If we spit out high concentrations of neurotrophins and stimulate P75, we can trigger apoptosis in that presynaptic neuron. And that's what we're seeing in these data. So in panel A, B, and D there, the green signal is showing you caspases. Caspases are the proteins that carry out apoptosis. These are essentially little molecular scissors. They're going to cut up all the rest of the cell, whether it's DNA, some protein, or the lipids. We're going to go and cut up cellular components. By we, I mean the caspases. So the more green we see in the cells, the worse. When you have activated caspases, the cell rapidly undergoes apoptosis. So we can see a few dead cells in part A, and that's to be expected. Not every cell makes it in cell culture. In panel B, they've added a high dose of nerve growth factor. So this is another neurotrophic factor. We discovered that you needed it for nerves to grow, so that's how it got its name. The red signal is showing us just the expression of P75, so that toxic receptor. What's more important to see is the increase in green in panels B and D, and that's what happens when you have high doses of NGF or BDNF, respectively. So with that high concentration of neurotrophins, we stimulate P75 and we activate caspases, triggering apoptosis. We know that caspases play a role here because they do, and because of panel F. So if you look in panel F, the higher that bar, the more, or the greater number of dead cells they saw. So start off with control, not a lot of dead cells. There's panel A. That next bar, pretty high, that's panel B. Lots of dead cells because we put on too much nerve growth factor. If we put on that same high dose of nerve growth factor, but we knock down caspase expression, not as many dead cells. The fourth panel there is just an important control where they take the same knockdown vector and scramble it up. Don't worry about it. Just look at those first three bars there. And really it's just the first two that matter. If you add a bunch of neurotrophic factor, either in culture or by having hyperactive neurons, and they spit out too much, that stimulates P75 and we get cell death. And cell death is a pretty quick process, so this movie is showing us apoptosis. So here we're looking at some cultured cells. The red is showing us mitochondria, and inside of them we have this green fluorescent protein that's going to show us whenever we have stimulated caspases. So you'll see the cell turn green whenever the caspases are turned on. They're going to trigger apoptosis, and you're going to see a rapid transition right there. And the cell quickly goes from alive to dead, and you'll see these little bits of membrane break off. This is a very controlled process, so the cell destroys itself in an orderly manner to prevent inflammation. This way we don't have to go back to main point 4. We can instead move on to the review. So have a look at these before class. Think about anything that doesn't make sense. Fill out the question boxes on the class website. and I'll see you later.